Hi, my name is Phillip Monte,
And I'm Alondra Soto.
And my name is Ilya Makushin.
and we're all three students at the University of South Florida, and we're taking modern biomed technologies.
We are researching molecular dynamic simulations of cell membranes with the lipid POPC and comparing the results to experimentally known values.
The purpose is to show that simulations are accurate in modeling cell membranes and to assist biomedical engineers in developing future technologies and improve on drug delivery methods at a lower cost.
Cell membranes play a vital role in protecting cells and regulate the transport of materials and ions. Phospholipids form the basis of cell membranes and have two components, a hydrophilic head and a hydrophobic tail. The physical properties of lipids can change with temperature and can be found experimentally or through a relatively new process known as molecular dynamics. Molecular dynamics is a computational simulation method that models the movement and interactions of atoms in a molecule. Current research on cell membranes require expensive laboratory tools that can cost in excess of $100,000, with additional costs to use the lipids, dyes, and tracers. Dynamic simulations require a supercomputer cluster, which can be found at most research institutions and have low or no cost to access.
For the design, a GRO file, or graphic object bitmap, gives the Cartesian XYZ coordinates of each atom located in a 100 POPC lipid membrane section. This file can be used in a program called GROMACS, which derives Newtonian equations for each atom in the molecule. There are three components involved for running a simulation: energy minimization, equilibration, and the production run. Energy minimization finds the optimal orientation and arrangement of the atoms in POPC to give the minimum in atomic forces and energy. The purpose of this is to find the most stable arrangement of the molecule in nature. During equilibration, GROMACS takes the molecule through low and high energy states to thermally equilibrate it. This process is known as thermal ramping, which increases the temperature and then decreases the temperature after a few picoseconds. Essentially, it creates domains and constraints on the simulation of POPC and ensures the modeling is accurate. Production run is the final simulation step for the lipid bilayer POPC, which is ran for 100 nanoseconds to allow the atoms of the molecules enough time to interact with each other. This program was ran through the USF Computer Cluster, CERCI, which took approximately 10 days to complete. GROMACS gave output files for each of the four simulations, which gives the bilayer thickness and area per lipid. These values are then compared to the experimentally gathered values.
Now for the fun part, conclusion time. It was found from the simulation that temperature did have an effect on both lipid bilayer thickness and area per lipid. These results showed at most a 2% error for the bilayer thickness and a 6% error for area per lipid. This shows that molecular dynamics is fairly accurate in modeling lipid bilayers compared to experimentally gathered values. As of now, computers are not powerful enough to be able to model large-scale designs, but according to Moore's law, computational power is almost doubling every 2 years. With this advancement, it is plausible to assume that in the near future, computers could be able to simulate larger biological processes to assist biomedical engineers in developing new technologies and improve on drug delivery methods at a reduced cost compared to traditional experimental lab procedures.